Welcome back, class. In today's lecture, we're going to explore an important part of the field of microbial ecology. And we're going to do this by discussing the process of elemental cycling. That is the exchange of nutrients by microorganisms that makes life on Earth possible. This is sometimes referred to as environmental microbiology. We're also going to look at some of the more important tools and techniques that are used to study microbial ecology. We know already that microorganisms function as populations of similar organisms and as communities or mixtures of different microbial populations. And these microorganisms have evolved while interacting with each other, interacting with higher organisms, and with the physical world. They largely play beneficial and vital roles. When we talk about disease-causing microorganisms, they're only a minor component of the microbial world. Microorganisms, as they interact, also contribute to the functioning of ecosystems. And we know from living systems that ecosystems are self-regulating biological communities and their physical or non-living environment. Knowledge of these interactions is crucial in terms of understanding both microbial contributions to the ecosystems and also microbial roles in disease processes. But a major problem is in understanding microbial interactions is that only about 1% of microorganisms on Earth have actually been grown in the lab. The differences between observable and culturable microorganisms has been noted for at least 70 years. But more recent molecular analyses have really demonstrated the magnitude of the problem. Here are some definitions for you of populations, communities, and ecosystems. During their life cycles, microorganisms interact with each other in the cycling of nutrients, including carbon, nitrogen, phosphorus, sulfur, iron, and manganese. And this nutrient cycling, called biogeochemical cycling, when applied to the environment, involves both biological and chemical processes. And it's of global importance. Nutrients are transformed and cycled often by oxidation reduction reactions that change the chemical and physical characteristics of the nutrients. All of the biogeochemical cycles are linked, and the metabolism related transformations of these nutrients make life on Earth possible. Significant gaseous components occur in the carbon and nitrogen cycles, and to a lesser extent in the sulfur cycles. Soil, aquatic, and marine microorganisms often can fix gaseous forms of carbon and nitrogen compounds. In these sedimentary cycles, such as that for phosphorus and iron, there is no gaseous component. This figure illustrates nutrient cycling by microorganisms in purple and multicellular eukaryotic organisms in green, and also the abiotic chemical world. So all the biogeochemical cycles are linked with energy you can see coming in from sunlight, but also from pairs of reduced and oxidized compounds. Let's look at a couple of these, starting with the arrow at the top of the figure, which indicates the oxidized form of a compound and the reduced form of a compound. So if we look at the most oxidized form of carbon, you can see in the box below the pink end of the arrow, that compound is carbon dioxide. At the other end of the redox spectrum, we have the most reduced form of carbon in methane, CH4. What about nitrogen? Here we have the most reduced form of nitrogen, shown here as the ammonium ion, NH4+, and the most oxidized form as nitrate, NO3-. And of course, for many of the nutrients, there are intermediate redox forms. This table shows the major forms of carbon, nitrogen, sulfur and iron that are important in biogeochemical cycling. We can also see the reduced forms, the oxidized forms of each element, and also the intermediate oxidation state forms. So for example, the first element on the list, carbon. Is there a significant gaseous component present? Yes, there is. The most reduced form is methane, CH4, the most oxidized form, carbon dioxide. And the intermediate oxidation form is carbon monoxide. Nitrogen, once again, has a significant gaseous component. The most reduced form is the ammonium ion, the most oxidized form, nitrate, and then our intermediate forms, nitrogen gas, nitrous oxide, and nitrite, NO2-. Sulfur also has a significant gaseous component. The most reduced form, hydrogen sulfide, and also sulfhydryl groups in organic matter. The most oxidized form of sulfur is sulfate, and here we have some intermediate oxidation state forms, elemental sulfur, thiosulfate, and sulfite. There's no gaseous component in the iron cycle, and iron is found in its most oxidized form as ferric, in its most reduced form as ferrous. Just note that because the iron cycle lacks the gaseous component, this is described as a sedimentary nutrient cycle. So within the carbon cycle, carbon is present in reduced forms such as methane and organic matter, and in more oxidized forms such as carbon monoxide and carbon dioxide. Here we have a carbon cycle that may look very different to what you've seen in other subjects. Aerobic processes are noted with blue arrows to the right side of this figure. Anaerobic processes are shown with left arrows to the left. Now, even though carbon cycles continuously from one form into another, let's just say that this cycle begins with carbon fixation, which you can see just below the words anaerobic and aerobic at the top of the figure. And of course, you know that carbon fixation involves the conversion of carbon dioxide into organic matter. And you can see that this happens both aerobically and anaerobically to contribute to a common pool of organic matter in the centre of this figure. Plants, of course, are often thought of as the principal carbon dioxide fixing organisms, but at least half the carbon on Earth is fixed by microbes, especially marine photosynthetic prokaryotes and protists, for example, cyanobacteria and diatoms. Now, just looking at that common pool of organic matter in the centre, you can see a blue arrow linking this pool with carbon dioxide, and it's labelled respiration. There's also a red arrow linking this common pool of organic matter to carbon dioxide towards the left, and it's labelled anaerobic respiration and fermentation. So what this shows is that this fixed carbon within the pool of organic matter can be oxidised back to carbon dioxide through either aerobic or anaerobic respiration and fermentation. Now let's focus on the anaerobic processes in red, just for now. A red arrow links carbon dioxide on the far left with methane, CH4, towards the bottom of the figure, and it's labelled methanogenesis. A red arrow also links the pool of organic carbon in the centre, the organic matter in the centre, with methane at the bottom, and it's also labelled methanogenesis. So what these two arrows show is that inorganic carbon in the form of carbon dioxide and organic carbon in the form of organic matter can be reduced anaerobically to methane. And this methane is produced by various archaeans in anoxic habitats. Note also the role that hydrogen plays in this process. Hydrogen is a strong electron donor, and it's produced when organic matter is degraded, particularly under anoxic conditions when fermentation occurs. Now let's consider this methane, the CH4 at the bottom of this figure here, and let's consider it in terms of aquatic environments. So within a water column, the anoxic zone where methane is produced is often below an oxic zone. Therefore, as methane moves up the water column, it's oxidised before reaching the atmosphere. And we can see that indicated by the blue arrow at the bottom of this figure, and it's labelled methane oxidation. However, as you can imagine, in many situations, such as in rice paddies, without that overlying oxic water zone, the methane is released directly to the atmosphere, and thereby it contributes to global atmospheric methane. Rice paddies, ruminants, coal mines, sewage treatment plants, landfills, and marshes are important sources of methane. Now, in the carbon cycle that we just looked at, there's no distinction made between different types of organic matter that are formed and degraded. And this is quite an oversimplification because organic matter varies widely in terms of physical characteristics and in the chemistry of its synthesis and degradation. Organic matter varies in terms of elemental composition, the structure of basic repeating units, linkages between repeating units, and physical and chemical characteristics. So degradation of organic matter is influenced by a series of factors, and these include nutrient availability, abiotic conditions, so for example pH, redox potential, osmotic conditions and so on, and also by the microbial community that's present. This table summarises the major complex organic substrates used by microorganisms and also the characteristics that influence decomposition and degradability. Because microorganisms require each macronutrient, if an environment is enriched in one nutrient but relatively deficient in another, then the nutrients may not be completely recycled into living biomass. So for instance, we can see here chitin, protein, and nucleic acids contain nitrogen in large amounts. Now, if these substrates are used for growth, then the excess nitrogen and other minerals that are not used in the formation of new microbial biomass are released to the environment in the process of mineralisation. Mineralisation is the process by which organic matter is decomposed to release simpler inorganic compounds. So for example, carbon dioxide, ammonium ions, methane, and hydrogen. The other complex substrates listed in this table contain only carbon, hydrogen and oxygen. If microorganisms are to grow by using these substrates, they must acquire the remaining nutrients they need from the environment. And this is often quite difficult as the concentration of nitrogen and phosphorus might be quite low. So this inability then to assimilate sufficient levels of a nutrient will then limit the growth of a given population. So for example, in open ocean microbial communities, growth of many of the autotrophic microbes is often nitrogen limited. In other words, if higher concentrations of usable nitrogen were available, for example nitrate and ammonium ions, then the rate of growth of individual microbes would increase, as would their overall population size. Those nutrients that are converted into biomass become temporarily tied up from nutrient cycling, and this is sometimes called nutrient immobilization, as you can see on this slide. Now, most carbon substrates can be degraded easily with or without oxygen present, but this is not always the case. The exceptions, as you can see in this table, are the hydrocarbons and lignin. Hydrocarbons are unique in that microbial degradation often involves the initial addition of molecular oxygen. However, anaerobic degradation of hydrocarbons with sulfate or nitrate as electron acceptors can occur. With sulfate present, then bacteria belonging to the genus Desulfovibrio become active. Anaerobic degradation of hydrocarbons proceeds more slowly and only in microbial communities that have been exposed to these compounds for extended periods. Oxygen availability also affects the final products that accumulate when organic substrates have been processed and mineralised by microorganisms. So under oxic conditions, which we can see in the top half of this figure, that is under aerobic carbon use, oxidised products such as nitrate, sulfate and carbon dioxide will result from microbial degradation of organic matter. In comparison, under anoxic conditions, which are represented in the bottom half of this figure, titled anaerobic carbon use, reduced end products tend to accumulate, including ammonium ion, sulfide, and methane. These reactions also illustrate that the waste products of one group of microorganisms, for example the chemoheterotrophs, may be used by a second type of microorganism. So for example, chemoautotrophs during aerobic carbon use, or the methanogens during anaerobic carbon use. The degradation of lignin is also affected by the presence of oxygen. Lignin is an important structural component in mature plant materials and it makes up approximately 1/3 of the weight of wood. Lignin is primarily degraded by filamentous fungi, and it's a process that occurs mostly under oxic conditions. Lignin's diminished biodegradability under anoxic conditions results in the accumulation of lignified materials, and this includes the formation of peat bogs.